Hey everyone. In the last module, you learned about mitosis and the cell cycle and about their importance in cell growth and cancer development. In this module, we'll be taking a look at meiosis. Why is meiosis important? Well, for one, it provides a lot of the genetic diversity that drives natural selection and evolution. And it's also responsible for a large part of the reason why we're all different from each other, at least genetically. Now, in this first video of this module, we'll take a look at meiosis in detail, and we'll be addressing two questions. And our first question is, how does meiosis produce haploid cells with different genotypes? And our second question is, how are meiosis and mitosis different? Before we get into meiosis, let's review the relationship between chromosomes and DNA molecules and the relationship between homologous chromosomes and non-homologous chromosomes. During G1, which is of course before chromosome replication, each chromosome is made up of one DNA molecule. After chromosome replication, which occurs during S phase, each chromosome is made up of two DNA molecules. If the two sister chromatids are separated, they would become two chromosomes, and are still, of course, considered two DNA molecules. Homologous chromosomes are those of the same chromosome type, and they have the same genes. Chromosomes of different types that have different genes are non-homologous chromosomes. Let's compare the basics of meiosis and mitosis. Throughout this video, I'll write the differences between the two in yellow, so when you come back to it, you can see what they are. But meiosis and mitosis are similar in several ways. In humans, both processes start with a diploid cell. For this diagram, we'll use a cell with only one chromosome type. So this cell has two copies of each gene, is diploid, so it's 2N. In fact, it's a 2N equals 2 cell. Recall that this 2 means that it's diploid, or has two of each chromosome type. And this 2 means it has two total chromosomes. N is 1, because this cell has only one type of chromosome. The two chromosomes present in this cell are homologous chromosomes, and have the same genes. Before a cell goes through either meiosis or mitosis, the cell goes through G1, S, and G2, and goes through the same checkpoints that we discussed previously. The chromosomes are replicated during S phase. So now the cells have 4 copies of each gene and twice the DNA molecules. But these cells are still technically considered diploid, or 2N, since they have 2 of each chromosome. Here's one chromosome, and here is the second. Note that at this point, each chromosome has 2 DNA molecules, or chromatids. As you've seen, mitosis has 5 stages, followed by cytokinesis, which yields 2 cells that are diploid, each with 2 copies of each gene, so these cells are 2N cells. They also have the same genotype. Meiosis, on the other hand, is comprised of meiosis 1 and meiosis 2. So it has 2 cell divisions. Each cell division has the same 5 phases as mitosis, though some texts break them up into only 4. What occurs in each phase is similar, but with some important differences that I'll point out on the next page. The stages of meiosis I have Roman numeral 1 after them, and the stages of meiosis II have a Roman numeral 2 after them. Meiosis I produces 2 cells, with 2 copies of each gene, like mitosis does. However, after meiosis I, these cells can pause and go through a phase called interkinesis, which is basically G1 and G2 of the cell cycle. Importantly, these cells do not replicate their chromosomes before going through meiosis II. Meiosis II usually includes the same phases as meiosis I, but because the chromosomes were not replicated, when the cells with 2 of each chromosome divide, the daughter cells only get 1 of each chromosome, yielding 4 haploid cells. In this example, there is only 1 chromosome type, so the cells only get 1 chromosome total. But in humans, these cells would have 23 chromosomes, 1 of each type. These haploid cells would all have different genotypes, which is caused by the processes of crossing over and independent assortment, which we'll discuss on the next page and in later videos. Both of these processes occur during meiosis I. So, to summarize, mitosis produces 2 diploid cells with the same genotype, while meiosis produces 4 haploid cells with different genotypes. On the next page, we'll see exactly how this is accomplished. In describing each phase of meiosis I and meiosis II, we'll compare them to the corresponding phases of mitosis. Now, this page is going to get very complicated and may require some extra concentration and focus to understand, and you still might need to view parts of it 2 or 3 times. Also, I usually don't put this much content on one page, but I'm doing it here so we can compare meiosis and mitosis in detail. In this example, we'll start in G2 and consider a cell with 2 chromosome types, a large blue type and the smaller pink type. Since the cell has already been through S phase, we can see that these chromosomes have been replicated. We will follow 3 hypothetical genes, the A, B, and C genes. And the cell is heterozygous for all 3. In prophase of mitosis, the chromosomes condense, but they remain apart as dyads. So we see 4 dyads here. But in prophase I of meiosis, pairs of homologous chromosomes pair with each other, forming a structure made of 2 homologous chromosomes and 4 chromatids, called a tetrad. There are 2 tetrads in this cell. The formation of tetrads is called synapsis. Unlike during mitosis, non-sister chromatids, like these 2, can undergo crossing over during prophase I. Crossing over is when non-sister chromatids, which may have different alleles, exchange DNA. It's the first of 2 reasons why meiosis produces different cells with different genotypes. Let's see how it does this. Here is one of our tetrads as it appears before crossing over. Notice that both sister chromatids of each chromosome have the same alleles. During crossing over, both chromosomes break, and part of each is reattached to the other non-sister chromatid. If crossing over occurs between two genes, as it did here, between the A gene and the B gene, the two chromatids that crossed over can have a new combination of alleles on them. Before crossing over, we had two chromatids that had an A1 allele and a B1 allele, and two chromatids that had an A2 allele and a B2 allele. After crossing over, this chromatid now has an A2 allele and a B1 allele, and this chromatid now has an A1 allele and a B2 allele. During the rest of meiosis, which we'll get to in a bit, each of these four chromatids will be sorted into a different haploid cell. In this example, the two chromatids that did not cross over still have the same alleles. One has A1 and B1, and the other has A2 and B2. These chromosomes are called parental chromosomes because they have the same alleles as the parent cell. The haploid cells that inherit these chromosomes are called parental gametes, if they are in fact gametes. The chromosomes that now have new allele combinations are called recombinant chromosomes and are in recombinant gametes. This is one reason cells produced by meiosis can have different genotypes. Different combinations of alleles, here A1B2 and A2B1, are produced by crossing over. Can crossing over occur between sister chromatids, like between both A1B1 chromatids? The answer seems to be yes, at least in some species. But since the alleles on sister chromatids are the same, crossing over between them does not produce new allele combinations, so we are not as concerned with these crossovers. In 3 dimensions, a tetrad really looks more like this. The synaptonemal complex, made of RNA and protein, holds the two non-sister chromatids together and facilitates crossing over. Here, we see the formation of a chiasma, or a site of crossing over, which will eventually be resolved before the chromosomes are separated. We can see from this representation that both pairs of non-sister chromatids could cross over with each other, giving 4 recombinant chromosomes and 0 parental chromosomes for this particular cell. That would require a crossover here as well. But in this example, only these two chromatids crossed over with each other, so only these two are recombinant chromosomes. Through prometaphase and metaphase 1 of meiosis, the homologous chromosomes in each tetrad are held together at their chiasmata. In fact, the formation of chiasmata seems to be important for holding the tetrad together. Of course, this doesn't happen in mitosis because there are no tetrads. Each homologous chromosome is off by itself. During prometaphase 1 of meiosis, tetrads are bound by microtubules from both sides of the cell. But during prometaphase of mitosis, each sister chromatid of each chromosome is bound by microtubules from opposite sides of the cell. During metaphase 1, the homologous chromosomes of each tetrad line up on opposite sides of the metaphase plate. While during metaphase of mitosis, all chromosomes line up right on the metaphase plate. This difference in how chromosomes line up during metaphase and the difference in how the microtubules attach is the second reason that meiosis produces cells with different genotypes and mitosis does not. In mitotic metaphase, all chromosomes are lined up at the plate. Notice that the sister chromatids of each chromosome have the same alleles. So when they separate, each daughter cell will get the same alleles. One cell will get 1A1, 1A2, 1B1, 1B2, 1C1, and 1C2, as will the other daughter cell. During metaphase 1 of meiosis, unlike during mitosis, the homologous chromosomes line up next to each other around the metaphase plate. How the chromosomes line up and what microtubules they attach to is random. So these chromosomes and the alleles on them can move in either direction, which will result in the daughter cells having different genotypes because they can potentially get different alleles. This is known as independent assortment of chromosomes and is the second way meiosis produces haploid cells with different genotypes. Let's take a closer look at how this works. For this illustration, we'll assume no crossing over. Let's say we have a cell with two chromosome types, once again, a large one and a small one. The G gene is on the large chromosome, and the H gene is on the small chromosome. And the cell is heterozygous for both genes. After chromosome replication, prophase and prometaphase, there are two possible ways for how chromosomes could line up during metaphase 1. They could line up like this, with the big G chromosome on the left and the big H chromosome on the left, or they could line up like this, with the big G chromosome on the left and the little H chromosome on the left. If possibility 1 occurs, these chromosomes are going left during anaphase. So one daughter cell, when it gets to metaphase 2, is big G, big G, big H, big H. These chromosomes are going right during anaphase. So the other daughter cell, once it gets to metaphase 2, is little g, little h, h. If possibility 2 occurs, with the big G chromosome and the little h chromosome on the left, then the genotypes of the daughter cells in metaphase 2 will be big G, little h, and little g, big H, big H. Once these cells complete meiosis 2, we see that possibility 1 gives these two genotypes in the haploid cells, big G, big H, and little h, while possibility 2 gives these two genotypes, big G, little h, and little g, big H. Keep in mind that possibilities 1 and 2 are equally likely to occur. So any given haploid cell has a 50% chance of getting one of these chromosome combinations and a 50% chance of getting one of these chromosome combinations. In other words, they have a 25% chance of getting each genotype. So we can define independent assortment as the haploid cells having an equal chance of getting different combinations of chromosomes, and by extension, they have an equal chance of getting each possible combination of alleles. So this is how independent assortment produces haploid cells with different genotypes. During anaphase of mitosis, sister chromatids are pulled apart. They come apart because the cohesin that is holding them together gets degraded. But during anaphase 1, the sister chromatids stay together, and it's the entire homologous chromosomes that are pulled towards one end of the cell or the other. This occurs because a protein complex called shugoshin binds to cohesin and protects it from degradation. This allows cohesin to hold the sister chromatids together during anaphase 1. During telophase and cytokinesis of mitosis, the cell splits so that the chromosomes pulled to one side during anaphase are inherited by one cell, and chromosomes that were pulled to the other side during anaphase are inherited by the other cell. Again, because the same alleles are getting pulled to either side, this produces two diploid cells with the same genotype. By this point, the chromosomes have also decondensed and the nuclei have reformed. During telophase 1 and cytokinesis, the cell also splits, although the daughter cells have different genotypes as we've seen. The resulting two cells then go through interkinesis, which again is like interphase but without DNA replication. The cells then move into meiosis 2. During prophase 2, which I'm not showing here, the chromosomes condense again. During metaphase 2, the chromosomes line up at the metaphase plate as they do during mitosis, but now there is only one of each chromosome type. During anaphase 2, shugoshin is no longer bound to cohesin, which allows the sister chromatids to be pulled apart. During telophase 2 and cytokinesis, these are sorted into the daughter cells, which are now haploid. So, by the end of meiosis, we have 4 haploid cells with one of each chromosome type and one copy of each gene, all with different genotypes. In this example, the chromosomes with A1 and B2 on it, and the chromosome with A2 and B1 on it, were produced from crossing over. So, different genotypes are, in part, produced by crossing over. We see those different combinations of A and B alleles in each cell. So, crossing over produces new genotypes with respect to genes on the same chromosome, here the A and B genes. We also see that this cell with the A1B1 blue chromosome also happened to inherit a pink chromosome with the C1 allele, and the A2B1 blue chromosome just happened to also get a pink chromosome with the C2 allele on it. Over here, A1B2 was inherited with C1, and here, A2B2 was inherited with C2. So we see that the independent assortment of chromosomes is responsible for producing new allele combinations or genotypes with respect to genes on different chromosomes. Okay, so at this point you should be able to explain how meiosis produces genetically distinct haploid cells and be able to understand the many differences between meiosis and mitosis. If you don't feel like you can, as always, feel free to watch part or all of the video again, and remember to complete the learning guide as well. Now, in the next video, we'll take a look at how to count chromosomes as we proceed through meiosis. See you then.